Talk about some work I've done during my postdoc at Leeds, looking at how carbon dioxide concentrations in the atmosphere affect nutrient exchange between wheat and avascular mycorrhizal symbionts. So the bad news is that the way modern intensive agriculture works is really unsustainable, especially in how we add fertilizers, nitrogen and phosphorus especially. With obvious ecological problems of soil degradation, but more alarmingly perhaps, we've got a phosphorus shortage coming up. We get it from phosphate-rich rock, which is running out. And so we need new ways to improve phosphorus uptake efficiency. And there's a lot in the literature excitement about can we use mycorrhizas to help. And as Maya has nicely introduced them already, we're talking about avascular mycorrhizas here, which association between plants and fungi in the soil, enabling this nutrient exchange where mineral nutrients from the soil go into the plant that the fungi is acquired from the soil, pass to the plant in exchange for photosynthetic carbon. And they're incredibly common. They colonize more than 70% of all higher plants, including all cereal crops. And as a result, soils are rich in mycorrhizal inoculum. So where we plant these cereals, they're going to become colonized. But the problem is that mycorrhizas aren't always beneficial for their plant partners. And this graph up on the right here shows that you can have up to a 50% reduction in plant biomass after being colonized by these fungi. But on the other side, you can have a 50% increase in biomass as well. And so if we're going to use these in agriculture, we want to make sure that we're getting that benefit. And if possible, avoid this mycorrhizal parasitism on this continuum here that we've got. And plant genotype, we know, is a big driver of what causes this position on this continuum. And so that's what I'm going to focus on today. But we also know that soil nutrient availability and, as we've seen, fungal genotype can also affect this. We also need to take account of the fact that we're living in a changing environment, and CO2 is obviously a massive part of this and a theme through this conference. And there's a potential for this so-called carbon fertilization effect in mycorrhizal fungi, where elevated CO2 in the atmosphere allows greater carbon fixation by the plants, makes a stronger source in the plant so the hyphae can get more of this carbon, which should promote more hyphal exploration through the soil, more nutrient uptake, more nutrient transfer to the plants, and then the cycle continues, is the theory. Whether this occurs in crops, we're really not sure, so it needs testing. So we've got two main questions for our project. Do cultivar and carbon dioxide concentration affect mycorrhizal phosphorus uptake in wheat? And the opposite side of that, is fungal acquisition of carbon affected by cultivar and carbon dioxide concentration? And we address this with some experiments in the greenhouse. We've got 3 cultivars of spring wheat, Avalon, Cadenza and Skyfall, and we've grown those at 440 and 800 parts per million to represent current ambient and elevated CO2 concentrations. And we're growing them in a live farm soil, so it's not been sterilised, so there will be a resident mixed community of mycorrhizal fungi in there. But sorry, Marjorie, we have inoculated with Rhizophagus irregularis, which we thought was very common, but maybe less common than we thought. And we grew these for 11 weeks to flowering, and then to quantify the trade between fungi and plant, we've used isotope traces of phosphorus and carbon. But before I move on to the isotopes, I'll just talk about fungal and plant growth. On the left-hand side, you've got a graph showing the 3 cultivars, Avalon, Cadenza and Skyfall, and how much of their root has got fungus growing inside it. And you can see that it's all pretty high. We're averaging high 50s to 70% of the root length has got this mycorrhizal hyphae growing in it. And there don't seem to be many strong trends about how cultivar or CO2 drive this. Now moving across this graph on the right, you've got phosphorus concentration in the shoots of these 3 cultivars, and we do see some variation here. So Cadenza has got higher phosphorus than the other 2 cultivars, and especially at higher CO2, there's this CO2 effect improving phosphorus uptake in Cadenza. Is this due to the phosphorus coming through the mycorrhizas, or is this just the plant uptake? This is where the isotope comes in. So in each of these plant pots, we've got 2 plastic tubes which have got windows cut into the sides and then a mesh wrapped around it and sealed on. And the mesh is fine enough that the roots can't grow in, but the mycorrhizal hyphae can. And so we create this mycorrhizal hyphal access compartment. and we add the phosphorus isotope into there, and then we can trace its uptake into the shoot material. And that represents that mycorrhizal fraction of uptake compared to roots. And to control for any diffusion of phosphorus out of there, or microbial cycling and then diffusing out and then root uptake, in half of the microcosms in our control treatments, we add it to this core that we rotate. And so it severs those, it severs those mycorrhizal hyphae. And so the conductivity from inside the core to outside is cut. And so then 10 days later, we harvest all the biomass and we compare the amount of phosphorus in the shoots from the access to the no access treatments, and that gives us our fungal contribution. And what do we see? Well, not much going on in cadenza, so suggestion that maybe the fungi aren't very active there at all, so possibly can't explain that difference in CO2 concentration in phosphorus in the shoots. And also in Avalon, not much fungal acquisition. But in Skyfall, and you notice the huge error bars on this spread of data here, we do have a lot more uptake, and also notice the log scale. And so it's looking like Skyfall are getting much more phosphorus through their mycorrhizal partners than either of the other two cultivars. And perhaps this is encouraging. It seems that mycorrhizas might provide similar or greater benefit to their plant partners in the future, CO2, with the caveat that this is very dependent on cultivar. And how about in the other direction? What's this trade for carbon going to the fungi? Using the same experimental systems, we enclosed them just before harvest, 24 hours before harvest, in a plastic labelling chamber. And then we add a pulse of 14 CO2, which, again, in the reverse direction, will be fixed by the plants, transferred down to the roots, and then into these plastic cores. And if we compare, at the end of the experiment, the amount of 14C in here to in here, subtract one from the other, we can then look at fungal acquisition of carbon. And when we do that, we don't see much trend at all. So we are seeing transfer of carbon, certainly, to the fungi, but it doesn't look like cultivar identity or CO2 concentration really affect that at all. I did say, though, this is only a 24-hour labelling period, and because these plants represent the only source of carbon that these fungi have got, we can look at the fungal biomass in the soil to give an indication over a longer time period how much carbon these fungi are assimilating. And when we look at this, we do see stronger trends. So Skyfall has got by far the least amount of biomass of fungal tissue in the soil, this hyphal length density. Cadenza is greater and Avalon greater still, and we also see differences in CO2 concentrations, although I'd say less strong than the cultivar differences. And if we go back to that previous graph I showed of phosphorus uptake via the mycorrhiza, you can see this disconnect between the amount of phosphorus that Skyfall is getting from its fungi and the amount of fungi that Skyfall is supporting. So it seems like it's getting the most benefit off the smallest amount of extra radical mycelium in the soil there. And what's more, it's doing it with similar amounts of fungi in the roots as well. So it's a suggestion maybe that the fungi that this Skyfall cultivar is associating with, something about this cultivar means that it's got a more efficient phosphorus uptake mechanism through its hyphae. And so, to summarise this part of the talk, did cultivar and carbon dioxide concentration affect mycorrhizal phosphorus uptake? Yes. And we can see that the CO2 effect varies by cultivar. And was cultivar acquisition of carbon affected by CO2 concentration and cultivar? Yes. And cultivar seems a stronger driver of this trend. And yeah, the CO2 acquisition was not linked to mycorrhizal uptake of phosphorus. And it got us thinking that mycorrhiza, very tentatively, we're going to suggest that will contribute phosphorus now and in future CO2. And we started to think how we can maximise this benefit, and the obvious thing to us was to explore the cultivar variability. And it's not something that we came up with independently. This idea has been in the literature for quite a while. There's a quote from a review paper in 2002 saying that these fungi are ubiquitous in agricultural systems, and the potential for reselecting crops that are more responsive is great. So our idea was to explore the wheat genome for what we've suggested are called mycorrhizal benefit genes. Is there something in there that means that Skyfall was getting more phosphorus than Avalon or cadenza? And so in collaboration with a trials company, ADAS, and the University of Sheffield, we've got this N8 pump priming grant to investigate and carry out a QTL screen. And this is possible because there is a mapping population of Avalon and cadenza crosses. So we grew 99 lines of the Avalon cadenza mapping population, and very simple phenotypic data. We've inoculated, and we're going to compare that against non-inoculated plants. So you take away the biomass in your inoculated from your non-inoculated, and you get your mycorrhizal growth response. And we use that in the QTL to identify these mycorrhizal benefit genes. We've inoculated with a mixture of 5 species of common mycorrhizal fungi here, and we grow in a very simple system of sand and perlite on a minimal media feed. And then we grew them for just 5 weeks and then harvested. And to our relief, after all that work, we find huge variation in the response to mycorrhizal colonization. This is in shoot biomass. And so you can see all the different wheat lines across the bottom here. Each bar is a different wheat line. And some of them show a 50% reduction average in shoot biomass. And all the way up at the other end, we got almost 100% increase in shoot biomass. So huge differences. We passed this data on to ADAS, and the good news is that they have found markers which are associated with shoot weight, root weight, shoot height, root weight ratio, all these things. Unfortunately, they've told me that I'm not allowed to tell you what they are. We can't pass that on just yet. Mostly because they say we need to do validation studies and check that this isn't just one artifact that we found in this greenhouse with me doing the experiments. So we want to do validation studies and then get some seed breeding companies involved and hopefully do field trials again to see if this effect actually happens, because obviously when we're going to be growing crops commercially, that system is infinitely more variable and complicated than our simple experiment here. Okay, with that, I'd like to thank BBSRC and N8 for money, ADAS and University of Sheffield collaborators, the New Phytologist Foundation for putting on this conference and giving me the opportunity to speak, and everyone in the lab at Leeds, especially Katie Field, who's my PI, and all of you for listening. Thank you.